What is up, my friends? We are going to do mitosis notes today. Mitosis, again, is just a part of
I won't respond to that.
the cell cycle, the division portion of the nucleus. And so mitosis comes after interphase, including the G1, that's the G2 phase, that preparation period before you can physically divide. And mitosis has four stages. It has the prophase, metaphase, anaphase, telophase. You'll often see it as PMAT. And that's what we'll dive into. And you'll see in the PowerPoint as well as in your notes, there are great photos that go with every stage. And I've labeled them in the PowerPoint, and I really urge you to label them in your notes so that you will always know and remember what's going on. Not only is it written down, but you'll be able to see it along with a label. Prophase, our first phase, it happens in two stages, the early stage and the late stage. So in the early phase, this is really based on our chromosomes themselves. They're starting to coil and thicken and become those distinct X's with a centromere where they're attached. They usually double their length at this time. The nucleolus will completely disappear and the centrioles will begin to move apart. You'll see these little yellow, finger-like things coming off of them. Those are called spindles. Those are made up of microtubules. And so these will be really important in the coming phases in ensuring that we can create two new daughter cells. You'll also see around the nucleus, the nuclear membrane, that white is starting to break apart. Late prophase is when our nuclear membrane has completely dissolved and our microtubules, or those little yellow fingers, are starting to invade what was previously the nucleus. Spindles are completely formed, ready to get to work. The spindles are structures that will end up separating the chromosomes. And so during prophase, pairs of chromatids become attached to the centromere of the spindle, and the centrioles have moved to opposite poles. You'll see them, one's at the top like the North Pole and one's at the bottom like the South Pole. They have those beautiful microtubules, the spindles coming out and starting to grasp at the centromere of chromosomes. Metaphase is that second phase, and you'll still see that those centrioles are truly at those opposite ends of the cell. Spindle fibers are going to start pushing and pulling chromosomes, and they're going to line up the chromosomes at the center of the cell. You'll see this truly beautiful horizontal line across the center. And each chromosome is connected to a spindle fiber at its centromere. Anaphase, or the third phase, the centromeres are actually going to split and divide so that each chromatid is moving to the opposite side of the cell. So each chromosome splits and one goes to each side. Microtubules actually begin to shorten the centromere as wrapping them all back up as it pulls chromatids to each side. And by the end of anaphase, two ends of the cell, or the two poles of the cell, have equivalent, complete, identical sets of chromosomes. Again, big idea is we're taking one mother cell, dividing it to get two identical daughter cells. And then telophase, as you can see, this is very unique. We're starting to see a nuclear membrane and a nucleus beginning to once again form, that nice purple shade. The nucleolus is starting to return, and our cells will begin to pinch in, creating a nice cleavage furrow. Our own chromosomes are starting to let loose again within that nucleus. So take a moment, try and challenge yourself. There are 5 different phases, including interphase, so interphase, prophase, metaphase, anaphase, and telophase. Check and see if you can maybe identify some of these based on the photos in your notes packets. Here are the answers, not just some of them. Anything labeled one as in prophase, 2, metaphase, you see that beautiful line across the center of the cell. 3, anaphase, where we see this very stereotypical pulling of the spindles and chromatids back to each pole. 4, telophase, we're going to start seeing a split between our cells. And then 5, interphase, where we're just preparing. We're getting ourselves ready. That's that 90% of the cell cycle. 3 more photos. Try and practice again. Pause and take a guess. We have 1 metaphase and 2 anaphases, although in our middle photo, you can also see there's a lovely, lovely cell labeled in its metaphase uh, as well. So cytokinesis, you're probably looking at the photo to the right and saying, but it's exactly the same photo as in telophase. You'd be correct. Cytokinesis usually occurs at the same time as telophase. This is the division of the cytoplasm. And at the end of the mitosis, at the end of, you know, our nucleus splitting, 2 nuclei have been formed, and each nucleus has its own set of chromosomes. We now need to make sure that each cell has its own set of cytoplasm and organelles. So, cytokinesis in animals forms a cleavage furrow, which is pinching the cells inward until they are 2 separate individuals, and each new cell will contain its own nucleus and cytoplasm and organelles. Meanwhile, in plants, it's not really possible for our cells to just pinch apart because of the cell wall. Um, So in plants, we'll actually form a plate across the center of a cell between 2 nuclei, and it will continue to form until they are completely separate from one another. You'll see this in the photos on the left. These are fantastic photographs that display and depict this cell plate forming. The top one has an arrow pointing to that very light. You can kind of see it, the wall being built across with the 2 darker nuclei. And the photo on the bottom, each cell is green, and you'll see a dark blue nucleus. There's a nice red line coming between the two, and that is that cell plate forming. Mitosis will take between 30 minutes and 2 hours, depending on the cell type. Again, it's the process of taking one mother cell and creating 2 new daughter cells that are completely identical to one another and its mother cell. The cell it came from, the cell of origin. Now, the results of mitosis. If we're looking at a unicellular plant or animal, it's resulting in a brand new offspring by asexual reproduction. Same genetics, same everything, no diversity. In multicellular organisms like humans, it's resulting in the growth and repair of the organism. So we get a paper cut, we get in a car accident, we get road rash. This is going to repair our organs and our body systems. So the importance of mitosis is that these 2 new cells are exact duplicates because it's ensuring that the new cell is able to carry on the exact same functions as its mother cell. It would be horrible. It would be detrimental if our skin cells didn't reproduce exact skin cells. It would be awful if they produced blood cells, right? We need to make sure that every time our skin cells are dividing, they're making new skin cells so that they can close our wounds, help us heal. So, regulation of the cell cycle, we see this lovely anaphase on the left. Uh, Frequency will depend on the type of cell. So, skin cells divide frequently, liver cells can divide, but only on rare occasions like repair. And then, very, very specialized cells like muscle cells or nerve cells don't divide at all. That's like when people have paralysis, they damage their spinal cord or their nerves, they can't get those back. They usually suffer. That's a lifelong detriment then that you'll see because that can't repair like a cut can. So, some controls on cell division. So, when cells come into contact with other cells, they actually respond by not growing anymore. So, when an injury like a cut in the skin occurs, the cells at the edge of the injury will begin to divide rapidly. This is where we're going to see more of that 30-minute to an hour long mitosis cycle happening. When that healing process nears completion, that rate of cell division is really going to slow down, really slow down, because you don't want an abundance of cells. Our cell cycle regulators are proteins found in the inside and outside of a cell that regulate cell division. Um, some of these proteins are responsible for starting and stopping cell division. Other proteins seem to speed up or slow down that cell division process. And the proteins will send out signals that prevent excessive cell growth. This keeps tissues from disrupting one another. And so, this is really preventing us from having tumors, which tumors are just excess tissue built up in the body. And we'll actually take a few days and we'll look at how cancer cells differ from regular body cells. And so when cells are uncontrolled in their growth, our cancer cells don't respond to the signals and the proteins that regulate the growth of cells. So cancer cells are going to divide uncontrollably and form masses called tumors. And so if the cells in a tissue grow uncontrollably, the consequences can be very, very severe, right? Tumors can damage surrounding healthy tissues. Tumors have been known to cut off nerve to the rest of the body, nerves in the face, nerves that allow people to walk, right? um, Nerves that move to the brainstem. And so all cancers have one thing in common: the protein regulators have failed to do their job, and we're still trying to figure out what this really means. Cancer is one of the things that's most highly researched, and yet it's one of the things we know nothing about. So this is the end of our slides on mitosis. The next time you come back to our notes, we'll finish up with meiosis, or sexual reproduction of our cells. But we'll focus on mitosis now for the next few days. So please keep this in mind. Keep studying and reviewing these, and you'll become experts before making your stop motion videos.